I'm really excited to be here and give you this talk. So I'll just get right into it. So this is me after 5 hours of loading 34 Promethean flow cells. So I look really happy because all of them are green, so that means they didn't get any air bubbles in here. And so basically each flow cell gives you about 1 to 2 terabytes of raw data, and then that translated into approximately 5 terabases of nanopore metagenomics data. And so I'm going to be talking today about how I got here. So as you can tell by the title, the focus is the San Francisco estuary microbiome. And so I think a lot of you would probably know that there's millions of people that live in this area, and also a lot of wildlife and a lot of animals in the water. But because of the millions of people that live in this area, it's highly anthropogenically impacted. And it's because of the wastewater treatment plants and urban and agricultural runoff. And so the estuary is composed of these three bays, and then also the Sacramento-San Joaquin River Delta, which is really important for agriculture. A lot of people in California don't even know this, but the Delta actually supplies 2/3 of the drinking water to California, to all across California, because a lot of Southern California draws this drinking water from the Delta. And because it's a terrestrial aquatic interface, it's actually very central to element cycling. So that's carbon cycle, nitrogen cycle. And so one of the things of concern for this estuary is that because of all of these nutrients that are flowing in from the wastewater treatment plants and agricultural runoff, it has this really high nutrient loading. And when estuaries have this high nutrient loading, it can lead to eutrophication. And basically what happens is the algae grows, the water becomes hypoxic, and then it kills everything in the water. And so we're not quite sure why the estuary is not experiencing eutrophication, but it's one of the things of interest. And also, we want to prevent this so we can help manage the estuary. There have been a lot more harmful algal blooms in recent years. So in Lake Merritt and also the Bay, thousands of fish died because of this eutrophication. And so it was caused by this organism called Herasigma akashiwo. And so the city was trying to help manage this in the lake by installing fountains to help oxygenate the water. But this really indicates that we need to try to figure out what's causing these harmful algal blooms as we continue with climate change. So these harmful algal blooms are really difficult to predict because the conditions could be the same from one year to the next, and one year you might get a harmful algal bloom and one year you might not. These harmful algal blooms also disproportionately affect vulnerable communities, and then also a lot of the indigenous peoples in California have cultural activities in the water. They harvest plants, they fish, and so they get exposed to the toxins that these harmful algal blooms produce. So in terms of trying to manage what's happening with the nutrients, people have made biogeochemical cycling models, and there's this model called the cosine model, but it only has it only coarsely represents microbes in terms of phytoplankton, zooplankton, and it basically just has these organisms transform these nutrients in this way. We don't know which species are transforming the different elements, and so the goal of this project was to help use metagenomics to fill this knowledge gap. And it's very surprising that in this area with so many universities that there's very, very few metagenomic studies, and most of them are 16S studies. So like I said, this is an area where the biogeochemical cycling is central to this area. And just as an example of why we really want to get into which species are important, parts of the nitrogen cycle that happen anaerobically, most of this is controlled by microbes. And so we really need to understand who's there and what they're doing. The other part of this that's really important is understanding the role of viruses, which are often ignored. So a lot of times the viruses will cause what's called a viral shunt. So when the viruses lyse the cells, they actually increase the nutrient cycling because they release all of the nutrients that are in the cells. So that was another goal of this project. So I think most people here will understand how difficult shotgun metagenomics is, but I just wanted to emphasize a few points. So currently, these MindMag standards for rating metagenome assembled genomes as high, medium, or low. High is 90% complete, medium is 50% complete. So this really indicates how difficult it is to really assemble a complete genome from the metagenome. And this is a problem because we can't really know what the full capabilities of an organism is, and you might have contamination from genomes from other organisms in your mag. And so the way to think about this is, if each genome was a puzzle, and then you dumped all the puzzle pieces into a box and then tried to reassemble each of the puzzles, that's kind of what environmental sequencing is like. But it's really worse because it's like throwing it into a blender and then trying to reassemble all the puzzles because of the way that Illumina sequencing works. And so what we did was we turned to long-read metagenomics. And so this is the exact same DNA sequence with Illumina and nanopore sequencing. And these are assembly graphs as a representation of what the connections are between the contigs. And so you can see with the nanopore sequencing, we're starting to get complete circular genomes out, and the tiny little highlighted circles are plasmids and phage that are popping out. And so we decided to apply this to study the San Francisco estuary. And we wanted to create this catalog of complete genomes to help, put into these models to help figure out what the levers are so we can help manage the estuary. And then it also, because you get a complete genome, you can also normalize by the genome abundances. And this was in collaboration with the Department of Water Resources and the United States Geological Survey. So you might be asking yourself, can we actually decompose the entire microbiome? And this is preliminary data showing that we think that we can. This was one nanopore sample that we did, and then we mapped the Lumina reads from this, the assembly from this Lumina study. And you can see that the similarity is actually pretty high. So we felt like that we could actually decompose the entire microbiome. So I'm running a little short in time, so I'm gonna go a little bit faster. So I'm gonna talk about this pilot study from this USGS station. And so for this, we optimized the DNA extraction because you can't get long reads unless you have long pieces of DNA. And then we also optimized the bioinformatics methods to refine our genome so they're more complete. So this is just an example of how our DNA extraction differed from the standard one from a kit. So environmental samples are much harder to extract DNA from because they have a lot of environmental DNA that are short fragments, as you can see here. And so this is also size selected, but you can see that the DNA is much longer. Another thing we tried to do once we got the sequencing was, can we classify every contig taxonomically classify each contig? And what we found out, and this is detailed in the paper, is that it also still relies on the database. We could do it. We got some complete picoeukaryotic genomes and also chloroplast and mitochondria. Here's some microscope pictures that we took. But also surprisingly, 40% of the sample was viruses. And so we started looking at the viral populations, and there's actually some strain-specific viral abundances that you can see here on the right. And a lot of these viruses were algal viruses that infect diatoms, which are in our samples. So it's not surprising, but it indicates that we can actually look at a population all the way from viruses to picoeukaryotes now in one sequencing run. So we also saw strains level differences in abundance for the bacteria and archaea. There was only one archaea in here. But this also allowed us to try to estimate the total number of species in the sample. And that also gave us an idea of how deep we needed the sequence in order to really decompose the microbiome, because you have to have enough coverage. And so that's why we did 5 care bases in nanopore sequencing, because we figured out how deep we needed the sequence based off of the diversity in our sample and the coverages. And so we have data from 8 USGS stations, and we're also getting complete genomes that have really been desired by marine microbiologists, such as SAR86 and SAR11. And if you thought that was a lot, we now also have 5 care bases of samples from the Baltic Sea, which is really exciting. And so I just want to thank everyone that was involved in this project, especially Torben Nielsen, who was with me in all the different steps, and also my collaborators at JGI, USGS, and DWR, and Northeastern.